Happy fall campers.
Happy fall. Yeah, it's getting chilly out there, at least up here in Northern California, anyways. How have you
Yes,
been? Long time no buzz,
it
right?
has been. I wonder why. Why has there not been a buzz for about a month?
Well, tell us, Keon, why?
Well, we're transitioning the buzz from going monthly to quarterly. So now it's seasonal. We're
Hmm.
gonna have a fall edition, we're gonna have winter edition, spring, summer editions. And this is gonna make it so we'll be able to include more information per edition. And so we're excited in being a little bit more full of content and look forward to sharing this first fuller version with you today.
Yeah, yeah, sounds like a plan. Okay, well, let's buzz through this part of the buzz anyways, because we have some really cool interviews at the end, don't we?
Yes, we do have a
Hmm.
couple of new interviews with new organizations.
Hmm.
Okay, let's start with some announcements. So we've had some requests in the previous classes on when the new schedule for 2024 for the classes to be posted. And that is going to happen in December. So check back in late December. It'll be posted on the class schedule on our website. That's what the page is called. And once it's posted, they'll all be posted. So the all of 2024 will be finalized and you can register for all the classes right at the beginning. And that's actually what we suggest you do. So go through, look at the ones you want to take, look at all the details. There's a link to the class catalog if you want to learn about the description. And
You know what I do? I just register for them all because then I get all the links to all the videos, right?
you get the calendar invites, correct?
Yeah,
Yes.
yeah, yeah,
And that includes the link to the Zoom. Yes.
yeah.
And if
Yeah.
you can't attend one of them, then if you're a member, a current candidate, then you can go into the video library section and just watch the previous recordings.
Yeah,
And yeah, you also, I see what you get the recording after the class too, correct?
yeah,
Yeah,
yeah. So makes it easy.
yeah.
CSBA is just around the corner, right? November 14, 15, 16. If you have offered to be a volunteer for a day in the camp, stay tuned. We'll be emailing you soon. And Kian, remind me how if I am a camp member and I'm trying to figure out when my year deadline is up for when I have to take my exam, how might I go about doing that?
Yeah. So your deadline is 1 year from registration. So the easiest way to check that is to check your email and find the registration confirmation email that was sent to you. And in there, look at the date. It might include it in the email depending on when you registered.
Mhm.
But just look at the date that was sent and it's 1 year from that date. It may also say in the email body. So
And
it
if I
depends
can't
on
find that, what
you
can I
can reach
do?
you can reach out to us and we
Oh,
will let you know.
okay. Thank you.
Okay, events corner. So we have certification program info session that was going to happen this week. We moved that to next week. So that's next Wednesday, 7 to 8 p.m. So if you're interested in learning more about the camp certification program info, then let register for that. And that is on our website under the certification tab. You can register for that session, and we'll be talking about all the ways that the program can support you in your beekeeping goals. And we'll be able to answer all the questions you have. And if you can't make it, then you can watch the previous recording. So you can do that. Or you could wait till after and watch the recording of it. But come and join us and ask any questions you have about the program. And this is intended for people who are interested in entering the program. If you're already in the program, you probably already know all this information, so it probably won't be too useful. So let's see what else we have.
Yeah, so it's going to be a ton of fun, November 11th, 9 to 12, online, introduction to mead, We've done this probably our 4th year now. And Mark Carlson, soon to be master beekeeper with the camp, always gives us a really in-depth learning experience regarding how to be a great mead maker. He's also invited Ricky Klein from the Groenfeld Meadery in Vermont. And Ricky will be offering the mead tasting. Mark will be running the honey tasting. So if you would like to participate in the mead tasting, please consider ordering the mead that you will be tasting from Groenfeld ASAP, because Ricky's going to need a couple weeks to get it to you. Shipping is free, and it's a great holiday gift and a ton of fun. Like I said, hope to see you there. And as I mentioned before, expect an email regarding volunteering for a day in the camp on November the 15th. And we hope to see you, even if you're not volunteering, at the Hilton in Orange County, Costa Mesa, for the 2023 California State Beekeepers Association Annual Convention. Always a good time, great to network, and there's a ton to learn. Yeah.
Yeah, no, and last year was my first year
Yeah.
there, and they had this trade floor set up, which we'll have this year too. And all these commercial companies and for large scale beekeeping, and also for smaller scale beekeepers too, for some of them. They show their products, they talk about their services, they have raffles. So the trade floor is a lot of fun. And then also they have panels and discussions as well. So if you want to hear from some researchers on the research they're doing, there's usually a few speakers on that. And part of the day of the camp is this year we're doing a panel, right?
Yeah.
So
Yeah.
that's going to be pretty exciting.
Yeah.
Well.
And we'll post details on our socials as we get closer. We're still waiting for the final schedule from the CSBA. And once we have that, we'll be sending all of our volunteers some info regarding what's happening on the 15th.
Fantastic. Okay, we have the final class in the new 3-part pollinator series run by Art
Ando. Yeah.
And so these classes have been very practical with the knowledge that Art has been sharing. He's talked about how to design gardens and how to really the practical methods and how to implement them, drip irrigation, everything you could think of in terms of purchasing and setting up gardens. And so we've had some great feedback. We hope you can join us for this final part in this series. And this will be on November 18th. Hope to see you there. And the final class that we will be offering in 2023 is technology in the apiary. So if you're interested in Internet of Things or interested in somehow monitoring your hives to aid in your inspections or in your beekeeping management in general, this class will go over the general tools and specifically some brands as well that have worked for some people, and how to implement them in your own apiary and how you can use and interpret that data to help guide your decision making. And doesn't have to be complicated, right? There's a lot of products on the market that are plug and play solutions. So there's a whole spectrum. You could set it up yourself from scratch and make it very custom, or you can have an already pre-built solution and still get the basic data if you go on vacation, for example, how hot your hives are, things like that. So you're going to get all the options of what you can do in terms of technology.
Yeah, our volunteers from OCB team, Benoit Poulequin, will be offering that.
Yes. And we have at the end of this year, as we do every year, the most fun event via Zoom, the certification program annual celebration. And we'll be excited to be talking through some awards to be given out, some acknowledgements of top volunteers, top marks that have been handed out, and we're gonna get to celebrate all the new knowledge that has been learned in the program and all the sharing that has been done in terms of learning as well. So I hope
Yeah,
you can join us for that.
yeah, yeah. It'll be a good time. Alina's going to join us as well.
Yeah.
Mm. Yeah, so I'm looking forward to watching these interviews. They're good ones.
Me as well. The first
Yeah.
one's with the Vannette Lab, and that is out of UC Davis, and we'll jump into that right now.
Okay.
Hi, campers. Now we have a new section here, an interview with an individual from the Vannette Lab. It is Dino Lorenzo Spartolacci. Thank you for being with us today.
Thank you for having me.
So, are you at the lab, or where is
Yeah,
that?
I'm
Oh
in the
yeah.
lab. It's we're located in the basement of Briggs Hall at UC Davis.
Okay. Okay.
Yeah,
So, would you say for the Vannette Lab, most of the activities are done out of the Laidlaw lab, or is it mainly done in Briggs, or what's the difference in the functionality and
yeah.
where things are done?
I think most of our work we do here at in the Vannette Lab, or we do out in the field. And for some people like me, that is out at the bee biology facility. For other people, that means going out to like Stebbins Cold Canyon, and other parks and stuff to actually maybe collect bees or flowers. Yeah, I often also go up to the Sierra Nevadas and collect bees.
Okay, wow. Yeah.
Yeah.
And so you specifically, your background academically, what is that? Like, what did you study at UC Davis, or did you study at another university, then come to UC Davis for employment?
I did my undergrad at Sonoma State University, which is not too far from where we are,
Beautiful.
Davis. Um, and then I took a year off after graduating from my undergrad. I majored in biology. I took a year off, worked a little bit, realized that I wanted to go back to graduate school, and started applying around. I knew that I wanted to study bees, or really insects more generally, and microbes, and kind of the interaction between the two. And so I started looking for labs that would kind of give me the tool set to pursue this research interest. I went pursuing that. I went to Madison, Wisconsin, UW-Madison, and worked in the lab of Dr. Garrett Sewin, studying rumen microbiology. Bovine ruminants, nothing similar to insects, except that I learned a lot about sequencing and kind of bioinformatics, and I really gained this tool set that I am now applying to insect microbe interactions, specifically bee microbe interactions. Uh, yeah, and after getting my master's at UW-Madison, where I went, I came here to UC Davis, joined the Vannette Lab, and I'm a PhD student now.
Okay. And just for my edification, what are bovine ruminants?
Yeah, bovine ruminants, cows, dairy cows is what we studied. Bos
Okay.
taurus is the scientific name. We worked mostly with Holstein dairy cows, which are kind of your classic like black and white, like black spotted cows. Um,
Yeah.
yeah, I was studying the ecology, the microbial ecology of their gut, basically. Um, but again, I was just really gaining the tool sets that I knew I needed to pursue my research interests in insect microbe interactions,
Okay.
more specifically bee microbe
So you learn like
interactions.
the methodologies of sequencing, all of the lab technical like procedures.
Yeah,
Okay.
exactly. I knew I needed to pick up, DNA sequencing and computational skills and stuff to like answer some of the questions that I was interested in. And so I spent my master's like equipping myself with that toolkit.
Okay. Thanks. Yeah, I'm just I'm not too well versed in gene sequencing, but you guys use PCR or use gel electrophoresis. I'm not sure if those are exactly what you use, but I remember from AP Bio those are what we were learned back then for
Yeah.
DNA, but yeah.
Certainly, we do PCR and run gels. That's a really good tool for looking at like presence absence of a particular like bacteria or microbe within a sample, whether it's a soil sample or a bee gut or something. You might, design PCR primers that are specific for a lactobacillus or a bifidobacterium, one of these bacteria that's present in your sample. Um, Perform the PCR, run the PCR product on a gel, check if you got a band. If you do, then that microbe is present. If not, maybe it's not present. Um, yeah, so that's kind of one of the basic tools that we use is PCR and running gel electrophoresis. I do a lot of sequencing, so I'll extract like all the DNA present in a bee or a honey pot or some other sample. I extract all that DNA and then I either amplify a particular gene region that's indicative of like bacteria identity. But I extract this DNA and then I sequence it and I use that information to infer the like microbial composition, like what bacteria are present in that sample. Uh, yeah, at what relative abundances are they there? Yeah, that type of
Okay.
stuff.
And so the work that you do that you just described, is that in general what the Vannette Lab works on? Or what is it? What would you say is like a quick two-sentence description of what the lab focuses on?
Yeah, I typically tell people we are a pollinator microbial ecology lab. We study pollinators, mostly bees, the flowers that they visit, and then also bacteria, and really how all these different entities, these different biological entities interact with one another. Um, how are bees moving around these microbes, these bacteria, when they visit flowers? How do microbes in flowers affect attractiveness of that flower to a bee, right? Like an increased pollination rates or something like that. Uh, yeah, that's kind of in a nutshell what this lab studies.
Okay. And in terms of like interesting studies, do you have any off the top of your head that you could describe in layman's terms in the time that you've been there? Yeah.
Sure. So when I first joined this lab, I kind of inherited a project examining the bacterial community associated with the gut of carpenter bees. Carpenter bees, I think a lot of people are familiar with them. It's a type of kind of solitary bee, although these bees that we were looking at are actually facultatively social, which is pretty interesting. Sometimes they can be social, sometimes they're solitary. And I inherited this project to characterize their microbial communities, their bacterial communities. And I found that they had features of their gut microbiota resembled uh, social bees in some respects and also resembled solitary bees in other respects. Um, Social bees have a very characteristic gut microbiota that's kind of like facilitated by their sociality, right? Like all the nestmates kind of interact with each other, they share food, so they have these like very similar microbial communities in their guts. Whereas like solitary bees don't have that type of behavior, and so they have very different uh, microbial communities in their guts. And the carpenter bees are like facultatively social, and they were somewhere kind of in between that in terms of like gut community, which is really cool to see. Um, so that's a project that I inherited when I first joined, and I think was really cool. There's other people in our lab that study other types of solitary bees, like not honeybees or bumblebees, but solitary bees, and the microbes associated with them. And yeah, there's other people that study kind of like how certain types of antifungal compounds affect the gut microbiota housed by bees and whether or not those gut microbes can protect bees from antimicrobial compounds like an antifungal. Uh, yeah.
Yeah. Have you guys done research? Does bacteria play a role in the production of honey? I know that there's many enzymes in the in the bee that help with that process, but are there also bacteria? Have you guys done research on that? I'm just kind of curious.
Yeah, I really don't know 100%. I don't really
Yeah.
know. Um,
Okay, new area of research, or
yeah,
maybe it's already been looked into, but
it
yeah.
probably, yeah, I imagine
Yeah, it
so.
probably has. Yeah.
Um, I know that like the chemical environment that is honey is pretty, I would think, pretty demanding, right? It's like a very, very high sugar content, and that is going to like automatically kind of shape what type of bacteria can live in it, right? You're going to have bacteria that are very tolerant of these high sugar concentrations. Um, Whether or not they play a role in actually producing it, I don't know. But the chemical environment of honey is definitely selecting for particular microbes that, um, maybe bees like in some way.
Mhm. Yeah,
Yeah.
another subject I'm thinking of is the immune system. And I know at the El Nino Bee Lab, they studied phytochemicals of flowers and pollen and how those can support the immune systems and the overall health of bees. Do and actually for humans, I remember when I was younger, probably around early 2010s, there was this huge outburst of research in high school. I remember reading all over Facebook about the gut microbiome of humans and how it's important in regulating health in general, immune system, and also your brain as well. The nervous system's all connected, and people with depression had a completely different type of gut microbiome. We're producing different, chemicals in the body that were affecting the nervous system, and eventually even causing depression or being a part of that. So, is there any research that you guys have done on the gut microbiome um, for bees?
Definitely. So I would say, like, I personally, that's like 90% of what I study is bee gut microbiome stuff. And a lot of people in our lab, that's very much what we study. Uh, Microbiome is kind of like a hot topic right now, and it has been. It's been increasing in popularity, which is great. There's a lot of like functions that are being ascribed to the gut microbiome. Um, as you pointed out, one of the maybe one of the most interesting for like humans and stuff is this like gut brain axis. The idea that like your gut microbes are producing compounds and stuff and yeah, influencing your nervous system, right? It's the gut's kind of like a second brain. Um, in regard to your question about how gut microbes and maybe microbes more generally can protect against
Immune
pathogens,
system, yeah, that was what I was getting at. Yeah,
maybe
thanks.
support the immune system.
Mhm.
uh, there's a lot of very cool research in this area. It's been shown pretty strongly in bumblebees, and I would imagine in honeybees, though I'm not as familiar with that literature. But it's been shown pretty robustly in bumblebees that certain gut microbes and gut microbes in general play a very important role in protecting bees from kind of invading pathogens. Specifically, I think it was Crithidia bombies, what like the like foundational research in this topic was looking at. And the kind of mechanism, the idea of this is that gut microbes are present within the gut at a very high density, right? These bacteria and fungi are present at very high abundances, and they are able to prevent invading pathogens from colonizing the gut through a variety of different means. One way is simply being there, right? Taking up space and using resources or preventing these transient microbes that might cause infection from being able to establish like a steady population. That's one mechanism. Another mechanism is gut microbes are changing kind of the environment of the gut, right? They might increase the acidity of a gut environment that then prevents pathogens from being able to invade, right? A pathogen might not do well under very acidic conditions. And so if you have gut microbes that are changing the environment to prevent a microbe from colonizing, great. Or a pathogen from colonizing, that's great. Um, it can be more direct too. Certain microbes can produce compounds that actually inhibit the growth, like kind of more directly of pathogens. So yeah, there's definitely research looking at the gut microbiome of bees, and how it improves the health of bees, and it's important for the health of bees. There's also research looking at how gut microbes kind of quote unquote train immune systems. That's an idea in like the human gut, where like your immune system is sort of trained by your gut microbes. I don't really know
It's
too
like
much about it.
possibly like part of your natural development where your body needs certain stimulus to develop the proper way. I know in other areas of I've learned that. So the gut microbiome, like in the healthy humans development, it needs to be there to push the body to develop in a certain way. Is that what you're saying, or
Yeah,
something slightly different?
I think there's research looking at this in mice.
Okay.
But if you have kind of germ-free mice, mice that is with like no bacteria in their gut, the actual morphology of gut tissue looks completely different than the
Oh, wow.
morphology of gut tissue with bacteria. When there are bacteria present, you get these like finger-like projections, these villi that form kind of within the gut, and like the actual like histology, the histological characteristics of like the gut lining in these animals looks like fundamentally different, plus or minus microbes. Yeah, so there's definitely like interaction, right, between bacteria, microbes, and their hosts, which makes sense. Like we've been living with microbes forever,
Yeah.
right? So of course there's going to be like some maybe like interactions and crosstalk between them over time, over evolutionary time.
Yeah, I would even say like they've been living with us for just short period of time for how long they've been
Yeah,
around. Um,
maybe that's the more appropriate way to look at it,
yeah,
right?
yeah, yeah.
We're not living with them; they're living with us.
Yeah. Also, what you're saying reminds me of like processed food and like how it affects our gut microbiome. And I remember reading that specifically in relation to depression, which we were talking about earlier. And
Uh,
so, like if kids are eating, for example, processed food that doesn't promote the typical healthy microbiome that humans have developed with, that food doesn't have the right proportions of macromolecules. You probably know better, tons of other reasons why I wouldn't support. Um, yeah, that possibly affects us a lot too. It's all connected. Yeah, actually, so zooming out a little bit, going back to the lab, how many people
sure.
work at the lab? Do you know?
Yeah. So I think we have four grad students currently, one post back, a couple of undergrads. We recently had two postdocs, but they have left to pursue greener pastures for the most part. Um, they're no longer in the lab. So yeah, there's like 5+ individuals right now.
And how long has the lab existed?
Ooh, I don't really know. I think Rachel is relatively new faculty. She got tenure pretty recently. I don't know. I can't give you a hard date on how long it's been
Okay.
here. I know I'm part of, I think, the third cohort of PhD students here. And your average PhD student is like 4 to 6 years, with some overlap in between these different cohorts. So yeah, I would say
So
at least
like at least a decade?
10 years. Yeah,
Yeah, at least a
probably.
decade. Yeah, so in terms of our members and anyone else who's watching this on YouTube, um, where would you suggest they go to look to learn about the research? They go on Google Scholar? Would they go on the Vannette website? Would they go Facebook, Twitter? I know some researchers like to post on Twitter updates, so I
Hmm,
guess X now. Yeah.
yeah. You know, I just read scientific literature, but I recognize that your average person maybe doesn't have access to a lot of scientific journals. A lot of them are actually like pay to read, basically, which is unfortunate. But there are free journals out there. I would maybe look for scientific literature published by Nature and Science, like these high quality journals, that are geared towards maybe the more general public. That way you get a lot of kind of research digested for you
Yeah,
and
and
present.
in terms of Vannette Lab specifically, that's kind of where I'm
Uh,
getting at. If
okay.
there, if, is there like does, for example, the lead PI or whoever it is, do they post on X, or do you recommend for us to go on the website to see what's most up to date for the lab? Or
Yeah. I'm pretty sure the Vannette Lab, we publish our like recent publications. We have a list of them on our website.
okay,
Um, Vannette
okay.
Lab. Let's see.
Yeah, no, I'll put the lab link in the description if anybody else would like to check that out.
Okay.
And any other relevant links that you and I discuss later, I'll also put in the chat
Okay.
for the members.
I don't use Twitter or X, so I can't really speak to that.
Mhm.
But a lot of my lab mates do, and I'm sure that they publish their research or maybe retweet uh, publications and journals and articles that they really enjoy. Uh, in the past, we in the Vannette Lab every year have kind of like each of us, each member has put forth like an article that came out that year that we thought was really cool. And Rachel Vannette then publishes those, or like puts out, I don't know, not like a newsletter, maybe like a newsletter, but like here's a list of cool articles that we thought
Oh, nice.
were
Okay.
cool. Yeah.
Good to
And I
know.
think that's housed on the Vannette Lab, maybe. I don't know.
Okay.
If you go to the Vannette Lab and you look under the Vannette Lab website and you look under the publication tab, there is kind of a list of all of our most recent publications.
Great, thank you.
Yeah.
Thank you, Dino. Thank you for describing your experience with the lab in general. Thank you for the work you do. It's very important, especially this like emerging field that's growing. And we look forward to keeping in contact with you and sharing with our members anything else in the future that you think would be interesting for them. And yeah, thank you.
Yeah, of course. Thank you for taking the time to talk with me. I love talking about science, especially bees and stuff. I'll talk about it for hours if you let me.
Welcome back. So let's just go ahead and jump into the next interview. And this one is with the Bruin Beekeepers, which is a partner organization of CAMP. And they have started their program just a couple of years ago and really come really far. So Wendy had the opportunity to go down there and meet them.
Yeah, it was a great week in Southern California a couple of weeks ago. So that's when I grabbed this interview.
Let's jump in.
Hey campers, here we are on the rooftop of the Life Sciences building at UCLA. It is myself, Wendy Mather, co-manager of the camp, with Ray Soto, who is the team lead at the camp satellite on the Life Sciences building at UCLA. Ray is the resident beekeeper, you could say, resident teacher, and all things beekeeping. Ray, what is today's temperature? It's a hot one.
It's a nice 90 degrees Southern California
Whew.
weather.
Yeah, so we were gonna crack open a few hives, but it's close to 2 o'clock in the afternoon. I don't think they'd appreciate it, and it's kind of loud up here on the roof. So I think what we're gonna do is continue this conversation in an air-conditioned classroom. What do you say?
Let's go.
All right. Hi again, campers. We're back inside. We're actually in the vestibule or staging area prior to going out onto the roof. So there are a couple of really cool things happening here. There is, as you see over my left shoulder behind Ray, there's an EpiPen hanging on the wall there. And there's also a closet filled with PPE because, let's see that PPE, yeah. Yeah, so when any of the engineers come up this way to work on the AC units, etc., Ray will have some kind of a sign stating that beekeeping is in progress, and they can choose to don a bee suit or just hang out here. Safety is priority one at UCLA. We're back, campers. Way cooler in here, thank you very much. Ray has a lot of really, in my humble opinion, a lot of inspiring initiatives that he has spearheaded here at UCLA. One of them is got the catchy title of Bee With Me Weekend. Ray, tell us a bit about the Bee With Me Weekend.
So that was a project started for the Bruin Beekeepers at UCLA for the purpose of offering an opportunity for the students to get involved in the apiary and around the apiary.
And the students have to be somehow involved with Bruin Beekeepers,
Right.
or the camp, right? Okay, so they got a little bit of intel around beekeeping.
Right.
Okay.
I usually have a list of things that need to be done in and around the yard.
Yeah.
So they get to choose what they want to do. If they want to be up in the roof in the heat, they can, or they can be in the nice classroom like in here and doing some job in here.
Wow.
So it's really up to them how much they want to do. But for camp, so for those students who want to do the camp program and want to enter the ambassador program, they need to give me 5 hours of volunteer time as a prerequisite to just sign up for the camp
Excellent.
and become ambassadors. After that, if they want to move on to apprentice, I'm going to require another 5 hours from them as a prerequisite before they do that. So by the time they're ready for the apprentice exam, they would have had at least 10 hours of in-hive experience.
Yeah, at least, that's good. And for students who are for the most part, pretty transitory, I mean, they're out of here almost annually,
It's a lot to ask
Yeah.
of them.
yeah, but many hands make light work, and you've created a community of beekeepers here. Yeah, that's awesome. I'm also really interested in the extension course that you have offered this past summer. Tell me a bit more about that, please.
That went fabulously. It was the first time any such course is offered at UCLA.
Yeah,
Because UCLA is known for its entomology or any animal science, no ornithology or herpetology. We just don't do that kind of work, which
Yeah.
is a bit sad. But hopefully the bees will make an inroad and be our inroad into
yes.
that future. So it went great. I had 9 students. These were professional students, not our kid students. I call them our kids. students.
Yeah, adult learners.
These were adult
Yeah.
learners.
Yeah.
And they had a fabulous time. And they each had their own assigned hive, so they had to take care of their own hive, which made it even better for them because now they had an interest. Now they had a buy-in.
And this was first time beekeepers forever. This is
Th-
their
these
first
were-
exposure.
Yeah, first exposure.
Wow. And this happened in the summer. And how long was the class?
A regular term, 11 weeks. 10 weeks for class and then a final week.
Wow.
And my final was the apprentice practical exam.
Wow. So the curriculum that was delivered was pretty much the apprentice study guide. You were able to take them through that?
What I did is the book was the Beekeeper's Handbook.
Yeah. Simon Terrell.
Right.
Yeah.
So that was the required reading.
Okay.
It was supplemented by videos from the National Honey Show.
Okay.
So I picked out specific videos there that would supplement the readings that they were doing.
Right.
And in our discussions for the chapters, I embedded all of that with the apprentice study guide from the camp.
Awesome.
So the presentation is mostly from the study guide. Because that just goes hand in hand with the book.
Excellent. So after 11 weeks, 12 weeks?
So after 10 weeks of reading and preparing, that 11th week is come in and do your practical exam, your final exam. Show me
And?
that you know what you're doing. And 9 out of 9 knew what they were doing.
Wow. Fabulous. That's commitment. They showed up and they decided
They
to
showed up,
learn.
they were great.
Yeah, terrific. Wow, congratulations. And I also know this is going to be Ray's capstone presentation for his capstone award. So note that will be uploaded. His presentation on his capstone will be uploaded to the camp website shortly. Yeah. So Ray, the other thing that I'm really jazzed about is the fact that every opportunity you turn into a teaching. So you're all about empowering the students that come through the doors here at Bruin Beekeepers and through the camp, through the UCLA satellite. Tell us also about how you've been able to safely and responsibly expand your apiary at a very low budget.
We get our colonies through a partnership that was established by Bonnie Benson, the university's Deputy Chief Sustainability Officer. She approached the university's vendor that takes care of all our pests, so from raccoons to rats to cockroaches. But their contract requires them to do a live bee removal,
Okay.
which takes a lot of resources from them to do.
Yes.
So our partnership with them looks like they do the work. They'll remove a colony from wherever it is, if it's a hive or a swarm, whatever. They do that labor, and they turn around and give us those bees.
Okay.
We then relocate them to the roof in our apiary. And so we get free bees through them.
Are those freebies, are they sometimes a little overly defensive
Oh,
perhaps?
they
because
usually
they're
are.
unmanaged?
We usually have to wait until we have enough funding to requeen them, but our first intent is to requeen them
Yeah.
when we get the chance. But it's
Yeah.
because we are a student-led, student-funded project, funds
Yeah.
are hard to come by.
Yeah, yeah, I hear you. Yeah.
And when we do get those bees, we have the bee response team that responds to us receiving those bees. Because
The bee response
the bee
team.
response team, and it's a volunteer. I offer it every term to whoever wants to do it, and it's essentially a text. Hey, meet me at the apiary in 15 minutes because we have bees coming. And so whoever responds, they show up. I teach them how to take the bees from the pest control people and put them into a deep and set them up in a way that we're hoping they'll survive and they'll take. It's not 100% yet, but we are getting better. We've already been doing this for 2 years, and not only have the pest control people gotten better about removing them in a more kindly, gentler way,
Yes.
which has improved our queen right ratio.
Yeah.
Because before we were rarely queen right,
Wow,
and now we're mostly queen right.
Wow.
When they bring us a colony.
awesome.
And we've improved our processes here of making sure that they stay put. So
Excellent.
that's how we've grown from 2-3 hives to now over 16 at any given time.
Yeah, and you get the opportunity to teach on the go, especially around installing packages and making sure colonies are queen right. You teach, I'm sure, about temperament, pest and disease management through all of that simple recovery of swarms here on campus.
Yeah, and it's a big story when they bring it to us because we can tell where they came from, because sometimes there's leaves in the little vacuum bags that, oh, this is what we should be looking for. Sometimes they come with comb, sometimes they come without comb.
Yeah.
Sometimes the comb comes with evidence of how they were doing, because
Yeah.
we've had small hive beetle larvae in comb sometimes, like, oh, well, this tells us a story.
Yeah,
So it's a great learning experience for everybody, because every time we get it, it's a new story, it's a different story. And sometimes we get that story from the pest control people who, well, we removed them from this and that place. Oh, okay. And so we can start establishing that story. And it goes with the hive. So we can go to any one of the hives and they have a backstory to them. Well, this one came from this location, and this is how they came, and this is how strong they are now, or this is how weak they are now.
yeah, and you're really great at ensuring everybody uses the hive record sheet, right?
Right.
Yeah, which is also really important to track all of the history.
Yeah.
Yeah, well, that's just fabulous, fabulous. Thank you so much for your generosity of time today, Ray. Be well, everyone. So thanks to everyone out there for your interest in just elevating beekeeping to the science-based level across the state. And if you're interested to learn more in how Ray is able to do just that here at UCLA on the roof of the Life Sciences building, please reach out. Yeah, connect. And I know I'll be at CSBA November the 15th for a day in the camp. Ray's gonna be there, so we hope to see you there too. Until then, Ray, thank you for your service.
Thank you.
Yeah, be well, everyone.
As always, we appreciate your feedback for these videos. You can go ahead and click on the feedback link in the description. If you have any feedback you'd like to share, we're continually updating and reviewing the feedback to see how these videos can better serve the public. And
Yeah.
with that, we'll see you in winter and
Be well.
well.